Okay. Good morning. My name is Hugo Gutierrez. I'm from 1PLM Research Lab. I'm a postdoc. Today, I will talk about our new module, CHARMM-GUI LBS Finder and Refiner, that is used for ligand binding site prediction and structure refinement. A little bit of background. In computational drug design, in the early phase, there are a few steps that we have to go through. First, we want to have a target two-dimensional structure of our target protein, preferably in its hollow conformation bound to a ligand. And then we can go through chemical database filtering. Chemical database filtering to filter the types of ligands that are that look more like drugs, using Lipinski's rule of 5, similarity search, etc. And when we have this library of ligands, we can go through structure-based virtual screening. And this is done to identify active compounds for a specific target from our chemical library using docking methods. Docking will predict the protein ligand complex structure, so the binding mode of the ligands, as well as estimate the binding affinity of the ligand to the target protein. After docking, hopefully we get some hits, and these hits can be validated using experimental assays, biological assays. For a virtual screening to work properly, it usually requires high-resolution target protein structures, and also they should be in their hollow conformations. For example, if you see this figure on the top right, on the left is MDM2 protein in its hollow conformation, and we see the surface representation of the protein, and the ligand is overlaid on this hollow protein conformation. And on the right is the same protein in its apo conformation. So when you overlay this ligand onto the binding pocket in its apo conformation, you see a lot of steric clashes between the binding site residues and the ligand itself. We and others have shown that when you use hollow conformations, it is significantly superior as compared to when you use apo conformations of protein targets for virtual screening. We also know that there are a lot of protein structures in the PDB library. There are about 180,000 structures there. But there are still some important protein targets that are implicated in diseases that do not have experimentally solved structures. For example, cysteine protease in Alzheimer's disease. So when you have a problem like this, you can use computational protein structure prediction tools to get a three-dimensional structure of your protein that cannot be experimentally solved. And if you have seen the recent headlines in Science, you probably have also seen AlphaFold, Google's DeepMind AI computationally predicted protein structure prediction tools that can solve three-dimensional protein structures with really good accuracy. So you went through this blind test prediction called CASP, or critically assessment of protein structure prediction, and it came up with really good results. So this means that these types of protein structures that are computationally predicted, or they're also called homology models, they are becoming more attractive for people in drug discovery to use these types of structure for virtual screening. But we still have a little bit of problem with this, because overall, the quality of these, some of these predicted structures tend to be around 2 to 10 angstrom RMSD when we compare to the experimentally solved structures. So this is where structure refinement tools can be important because in structure refinement, we aim to improve the accuracy of these predicted structures towards experimental quality. So to reiterate the objective of our module, it's called LBS Finder and Refiner. LBS stands for ligand binding site. First, as a finder, it can predict the ligand binding site on a query protein that is submitted by a user. And as a refiner, it can prepare simulation programs for a user to do structural refinement on their protein structure. There are a few of important steps that this module goes through. First, it uses Gelosa Search to predict the ligand binding site. I will go through what Gelosa is in the next few slides. And then we also provide top 10 templates from Gelosa Search, as well as top 10 clusters of the templates. And we offer 3D visualization of these templates aligned on a query protein for structure analysis. And last, to do structure refinement, we set up distance restraint potentials and MD simulation inputs to easily run these types of structure refinement. So Gelosa stands for graph-based local structure alignment. It is a local structure alignment that's based on geometry and physical chemical features of the ligand binding site. This tool was developed in our lab. What it does is that if you see these two structures on the graph, you have two ligand binding site structures. So one in green and one in red. Gelosa Search does structure alignment that aligns the structure in green onto the structure in red through iterative maximum click search. And at the end, you have the structure in green aligned to the structure on the left, where you're also provided with a GA score that ranges from 0 to 1, 1 being perfect alignment. Gelosa alignment depends on two important components. It's a normalized score from 0 to 1. Two important components include the physical chemical feature similarity between two different binding site residues. For example, two tyrosine residues, it's QI. And then you have DI, or the distance between a pair of aligned residues. So the more similar they are aligned, and the higher your GA score is, and the more similar the residues that are aligned, the higher your GA score is going to be too. The way we use Gelosa for binding site prediction on a protein is this way. So we have the whole protein shown in green ribbon diagram representation. Through Gelosa Search, we have curated a PDB structure library that contains these local ligand binding site structures along with their ligands. Gelosa Search aligns these binding site structures onto a whole protein structure until you get a maximum GA score. So the site that it aligns best would be selected as the along with its GA score. And top 10 binding site that templates that are aligned to a query protein are used to predict a binding site on a query protein. This is the general workflow of this module. What I mean by the PDB library is that we have a ligand binding site structure library that contains at least one protein and one ligand with resolution better than 3.5 angstrom. In total, we have about 41,000 structures, non-redundant structures. So for example, this protein, you have its binding site with stick representation in green and the ligand shown in pink. We have about 41,000 of these in our Gelosa library. And our definition of ligand binding site residues are protein residues that are within 4.5 angstrom of bound ligand in the crystal structure. In here, you can submit a protein that can either be a computationally predicted protein structure or a homology model or an APO structure. When you submit the structure, it will be processed through PDB reader to prepare the structure for Gelosa search. Since Gelosa search is a computationally extensive procedure, since we have to align more than 41,000 structures on your query protein, this job is submitted to CPU job scheduler. And there we aligned all of these binding site templates on your query protein. The templates that come up with better than 0.6 GA scores are collected. We do this because we have shown through our benchmark studies that when a template aligns to a local structure on your protein and it has GA score greater than 0.6, it is statistically significant that the two local structure are similar. After Gelosa search, we rank top 10 templates and display them in a result page for users to analyze the templates on their query protein. And user can select one or more templates for us to derive restraint potentials and to prepare MD simulation inputs in 5 different simulation programs for structural refinement, including NAMD, GROMACS, AMBER, OpenMM, and Genesis. Here is a display of our front page. On the left, we have a little bit of the introductions and overview of what this module does. And on here, you can upload, you can fetch PDB from RCSB of an APO structure, or you can upload a homology model. The structure will be prepared through PDB reader. And then once that's done, you can view your structure to examine that everything is okay. And then it can be submitted for Gelosa search for local structure alignment. Here is a graph that estimates the running time for Gelosa search. On the x-axis, you have the number of protein residues, and on the y-axis is the time in minutes. So for example, when your protein has about 200 residues, it takes about 60 minutes for Gelosa runs. You can add your email address on this box to be notified when the job is done. And also, you can retrieve your job through this job ID number, which is a unique number that for every job you submitted through CHARMM-GUI that can be retrieved using job retriever. This is a sample of the result page for LBS Finder and Refiner. We display an NGL viewer of your structure. In this case, this structure was from PDB, 1 PDB. And on the right-hand side, we have a table that contains top 10 templates that were aligned using Gelosa search onto this protein. There are 2 tabs on this table. We have top 10 templates, and we have top 10 clusters of the templates. And on this table, we have the PDB ID of these templates, the ligand RCSB ID, sequence identity between the template compared to the query protein, TM score that measures global fold similarities between the template and the query protein, as well as GA score or Gelosa score that measures local structure similarity between the template and the binding site residues that it aligns to. And when you select one of these templates, you can view the binding site residues as well as the ligand from this specific template on your protein. So in here, we selected this template 3DL5 along with its ligand DHF that is aligned to your query protein. And when you clicked on the PDB ID, you can view the equivalent residues that the template binding site residues are aligned to. On this list, on the left column are the list of residues that the template residues are aligned to, and S2 are the equivalent template binding site residues. So for example, residue I7 on your protein is equivalent to valine 9 on the template. And if you want to see more information about the ligand, you can hover your mouse to the ligand ID. The sequence identity informs you how similar your protein is to the template binding site from Gelosa. And the TM scores can inform you how similar the global fold between this template protein as compared to your protein. And TM score ranges from 0 to 1, with 1 being perfectly similar or the same. And GA score measures the local structure similarity. So as you can see, for some of the templates in this library, they can have global structures that are not very similar, about 0.3, but they do have very similar local structure or their binding site structure, ligand binding site residues. And on this table, since we display more than just one template, you can also select other templates on this table. For example, if you select template number 10, it will display this NADPH that also binds at the neighboring site on your protein. It's a cofactor to this ligand. And you can also display their equivalent residues. And on here, if you want to look at the types of interactions that the ligands are having with your binding site residues, they are also displayed where you can have hydrogen bonds, salt bridge interactions, etc. So in here, we show top 10 clusters that is done to reduce redundancy in the types of templates that we display on our result page. So for example, if you clicked on template 1, 3, 4, 5, 6, 7, 8, they are all aligned to this same or similar vicinity of binding site on this protein. When you click on the second tab for the top 10 clusters, all of these templates are clustered together, and the template with the highest GA score, or 3DL5, is used as a representative template in the top 10 clusters. So using the top 10 clusters, you can probably view more different binding site templates on your protein. So that was the general overview of how you can find binding site on your query protein. So now if you want to do structural refinement on that specific binding site, you can use this equivalent box to display the equivalent residues on your query protein that's shown here in cyan as compared to the binding site residues from the template that are shown in gray. So for example, when we look at the two different binding sites, the one from the template in gray and the one that's from your protein in cyan, we can see a few binding site residues that have different conformations. For example, this residue glutamine 37, which is equivalent to serine 37 on the template uh, binding site. They have two different conformations. The glutamine 37 is facing the ligand on that comes from the template, and it results in this steric clash with the ligand. But the one on the template is facing a different direction that is more amenable for the ligand to stay at the binding site. What we want to do in structural refinement is to change the conformations of your residues in cyan so that they can mimic the ones that are seen in gray. And this is done because if you remember from the introduction page, when you have holo conformations, they tend to be better for virtual screening or ligand docking as compared to when you have apo conformations of your query protein. The way we do this is we use harmonic distance restraint potentials. So on the panel up here, you have your binding site residues shown in green stick representations. And on the left, yeah, on the right, you have uh, this binding site as well as the equivalent binding site residues from the template that's shown in magenta. What we want to do here is we want to change the conformations of residues in green towards the one seen in magenta. We do this using harmonic distance restraint, where K is the force constant, Rij is the distance between atom I and J in your query protein, and R0Ij are distance between atom I and J in the template protein. And we can do this in two ways. One, we can just do it on the alpha carbon of those binding site residues. And another one we can do for both the alpha carbon of those binding site residues and the center of mass of the side chains. From this specific example that you saw for that protein 1 PDB, we saw that residue Q37 side chain was facing the inside of the binding site, and we want to improve that side chain conformation. So we would use the second option for that. Um, After you do this, CHARMM-GUI solution builder will prepare the protein in solution and input files for MD simulations, where you can select up to 5 different simulation programs, including NAMD, GROMACS, AMBER, OpenMM, and Genesis to run your simulations. And you can also use this option for hydrogen mass repartitioning, or HMR, that can accelerate your simulation up to 4 femtosecond in time steps. So for our benchmark tests, we use 2 different data sets. One is DUDE dataset. This is done for computationally predicted structure or homology models. DUDE stands for Directory of Useful Decoys and Hands. This is a set of protein targets that are diverse, including kinase, protease, nuclear receptors, etc. We selected protein sequences from this list where the number of residues are less than 300 amino acids so that we can run a quicker protein structure prediction method. We get the protein sequences and we run it through iTaser which is a software that's used to predict three-dimensional structure of a protein using protein sequences. And for this benchmark targets, we exclude templates that have more than 30% homology to the template, to the sequence, protein sequence. And then we get 48 homology models that we use for our module to do structure refinement. For benchmark number 2, we select apo crystal structures. And these proteins come from Aztec's diverse set, which is another data set that contains diverse number of protein ligand complexes, where we select the proteins from this list that have apo counterparts. And there are 20 structures from this list that we use for ligand binding site structure refinement. This is the benchmark results for ligand binding site structure refinement. The one on the left is from the UDE dataset, and the one on the right is from Aztec's dataset. The structures that we have are categorized into 3 groups. So the structures in group 1 are the ones that have ligand binding site all heavy atom RMSD less than 2 angstrom, meaning they are a little bit similar to the experimental hollow structures. And then group 2, they have 2 to 3 angstrom RMSD to the experimental structure, and group 3 have more than 3 angstrom. So they are the ones that are more different to the ones seen in experimental structure. For this data set, we show 88% success rate, where 42 out of 48 targets show improvement. And the average improvement is 0.95 angstrom, going from initial value of average of 2.94 to refined of 1.99 angstrom, which is about 32% improvement. And the improvements are quite similar across 3 different groups. Group 1, we have about 30% improvement, group 2, 38, and group 3, 30%. So it shows that regardless of the different types of binding site structures, we can still show improvement using our method. And for this, we want to generate hollow structure conformations for our binding sites, and we use apo crystal structures. We show 70% success rate. We have 40 structures that show improvement out of 20 structures. And most of these structures in this crystal apo structures, they fall into the group 1 category, where the apo structures show quite similar conformation as compared to hollow structure conformations. Because of that, we don't see a lot of improvements or a lot of differences before and after refinement. And this has been reported through a number of studies where the differences between apo and hollow conformations tend to come from the side chain orientations, but not so much as the alpha carbon conformations. And we also show, using our benchmark, we selected 5 representative structures, targets, and we use all 5 different programs to make sure that they are reproducible using different programs for structure refinement. And except for Genesis, which I haven't analyzed the data, the different programs show consistent refinement and similar results, regardless of what type of programs you use for the structure refinement. So in conclusion, we want to show this new module called LBS Finder and Refiner on CHARMM-GUI that a user can use to predict ligand binding site on their protein if they don't know where the ligand binding site is, or if they want to see if there are possible different sites that a ligand can bind to. Because if you see in our result page, we display top 10 templates instead of just one. And we also have the clusters of templates. So it gives a little bit more freedom for the user to analyze the types of local structure that might be similar from PDB library templates as compared to the structure, query protein structure. And these templates can also prepare distance restraint using 5 different MD programs for structure refinement. So what I mean to say is that in our result page, we provide a rich and interactive option for user where all of these top 10 aligned templates have information about their TM scores, the global similarity between the templates and the query protein, the local similarities to GA scores, sequence similarities, ligand ID, etc. that I think can be really useful for a user to know what kind of ligand can bind to their local structure on their proteins and what possible binding sites they can explore more. And we show that using our benchmark results from 2 different data sets, we see improvements in these protein targets. And yeah, I think that's it. If anybody has any question, please. No questions?
So just a general question.
Yes.
So when you develop or test this module, what was your the hardest part to make things work here?
I think for this module, one of the more computationally extensive part is to run Gelosa Search. So because of that, for a structure you submit, we send the protein to the CPU clusters to run Gelosa Search because we have to align so many binding site templates on your protein. So that was, I think, the more, I guess, the more difficult part of implementing this module. And also I want to, sorry. I want to acknowledge the people who work on this project. Of course, since you mentioned the module implementation, Sanjun Park did the implementation of this module on CHARMM-GUI. I run the benchmark test, as well as Han did the benchmark test for different programs, NAMD, GROMACS, Genesis, etc. Yeah.
Okay. Thank you.
Thank you.